Thank you very much, Ali, for agreeing to give a Build a Cell seminar, and you can get started.
All right, thank you so much for having me, Kate. My name is Dr. Ally Huang. I'm currently the lead research scientist at miniPCR Bio, and today I'm going to share a little bit of my work from grad school to now what I do at miniPCR Bio in terms of how I've been applying freeze-dried cell-free technology to educational applications and how some of those projects have actually gone from the classroom all the way to space, which is really exciting. So the agenda for my seminar today, I'm going to give a little background on the development of the cell-free system that I created in grad school called BioBits, and how we apply that to teaching biology and biotech in the classroom. I'm going to give an example of one of the lab activities that we developed using BioBits, our BioBits Central Dogma Lab. And then kind of describing how it's integrated into the classroom, we're going to move into talking about how this cell-free technology has actually gone to space, talking about two different projects we have sent to space so far, the initial cell-free technology, proof of concept experiment, and then finally this year's experiment, which is actually going up in a few weeks, fingers crossed. I'm supposed to go to the launch on the 21st. We'll see if that actually happens, but actually using cell-free to synthesize phage in space as a project. As an aside, I do have the chat up here in Zoom. So if you guys have any questions during my talk, feel free to put it in the chat. I'm monitoring and happy to answer. All right, so something that I've been really passionate about my whole life is basically outreach and STEM education, just making science as accessible to as many people as possible. And I remember being in high school, and the classes that really got me interested were all the really fun chemistry labs or the fun physics lab you can do in the classroom. But when it came to biology, especially molecular biology, we were really limited. As you can imagine, for biology, it's really hard to see and understand what's happening, right? In chemistry, you're mixing chemicals together and it'll turn different colors, but in molecular biology in particular, it's a lot of moving small amounts of clear liquids around, and it's really hard to tell what's going on at the molecular level. If you have experience working in the lab, you know that the equipment involved can be really expensive. Things like incubators, shakers, ultra-low freezers to store reagents. Those are simply things that a lot of classrooms and schools can't afford. And in addition, the time and the expertise that it takes to maintain this equipment and run these experiments are often things that teachers don't have experience with or don't really have the bandwidth to bring into their classroom. So when I was starting my grad school career at MIT, looking around for labs to join, I saw this article released by the Jim Collins lab over at MIT talking about their cell-free technology applications, this idea of being able to express proteins without live cells and just applying to a lot of things like on-demand protein production or low-cost diagnostics. And that to me sounded amazing. It sounded like magic. And a lot of the work that the Collins lab were doing at the time was applying it to health-related applications, such as this. You may have seen this paper before. It came out in 2016. This idea of using the cell-free reactions freeze-dried on paper and then just dropping a patient sample onto that paper, rehydrating the reaction, triggering this whole hold reaction here, and giving a visual readout, yellow if you're negative, purple if you're positive, in this case for Zika, which was the health issue at the time. And so when I joined the Collins lab, I decided to combine, my interest in the science space as well as my interest in science education and outreach, and apply cell-free technology to education. Because to me, the arguments for using cell-free and other similar systems in these health applications is that it's cheaper, it's easier to use, and it's fast. which are all great for things in the health and medical fields, but also really useful things for teachers in the classroom. So during the course of my PhD and my thesis over at MIT, I developed BioBits, which was basically just applying these freeze-dried cell-free reactions specifically for educational uses. This was a collaborative project both at MIT, over at the Wyss with Peter Nguyen, at the IMES where Jim Collins was, and also with Mike Jewett and his grad student, who is now a professor at MIT at Northwestern at the time. We came together and developed this kind of prototype of different activities, right? So we were thinking, okay, let's use this freeze-dried system where all the students have to do is to add a solution to rehydrate this reaction containing plasmid, and we can have that plasmid express a variety of different proteins, right? Obviously, we could express things like fluorescent proteins and colorimetric proteins for a visual output. But, we really wanted to engage the students and get them really excited about science. So we generated some reactions that could express enzymes, that could catalyze reactions that generated smells to use that sense, or even enzymes that can cross-link hydrogels so students can poke them. And we even went into more synthetic biology demonstrations by having students play around with programmable synthetic biosensors. And I don't have time today to get into each one of these, so I'm just going to highlight a few examples. On one end of the spectrum, the easiest thing to do is to just have students express fluorescent proteins. Doing so, they can learn about transcription, translation, how DNA goes to RNA, goes to protein. And we even developed a really fun activity where students get a 96 well plate, each with freeze-dried reaction in there, and they have to plan out what plasmids at what concentrations do I add to each one of these wells to create the design that I want. So the pictures you're seeing here are actual designs made by actual high school students that we prototype these activities with. And you can see here, some people made rainbows, periodic tables, and this was a really fun, engaging activity that allows the students to express their creativity, express their artistic side, but also tie it into a scientific experiment. So that was kind of the more basic experiment. On the flip side, on the other end of the spectrum, we thought it'd be really great if some of the more advanced students could actually play around with things like biosensors for diagnostics to learn more about synthetic biology. And so a really common experiment that biology teachers do with their students, it's one of the original hands-on experiments they would do, is basically you take like a strawberry or a banana, you squish it up with some soap, you filter out the lysate, and then you precipitate the DNA out with rubbing alcohol, and the students get to pull this gooey DNA out with a toothpick, and they're like, cool, it's DNA. But that was the end of that experiment. And we thought, what if we could actually develop an extension on this where they take that DNA and actually do an amplification, they actually amplify the DNA and then apply it to a sensor, a toehold sensor, that using cell-free, that can detect specific fruit sequences. And so we were able to develop two different toehold sensors, one with banana that identified banana DNA. So, the GFP only gets expressed if the banana DNA is added to the reaction, and it doesn't, pick up kiwi or strawberry. Same thing with the kiwi specific toehold sensor. And yeah, that was really fun to develop, just like this whole range of activities. We were able to prototype them with a lot of local schools, get good feedback from teachers, and we actually published all of these findings. So if you're interested in reading more about all of these activities and details, we published all these open source, so teachers could also access them. So we have actually 2 articles published in Science Advances, August 2018, as well as ACS and Bio, March 2019, and we actually made the cover for both of those, and those are our cover designs. So if you want to look more into that, definitely check out those papers. Someone is asking in the chat, yes, we are using E. coli-based extract. It's just a crude BioBits, to make it as cheap as possible, is just a crude extraction from E. coli cells. And you can read more about that in these papers. So when I graduated, I didn't want to be done with BioBits. I actually wanted it to be something that was available beyond the prototype than it was in grad school. So started talking to a lot of people, and I discovered miniPCR Bio, which was a company, which is a company that is local to Boston, near where I was in MIT, and their whole mission statement was completely aligned to mine. The whole idea of making science as accessible to as many people as possible. The reason that they're called miniPCR is they started out with low-cost mini PCR thermocyclers, like just this big. Actually, I have mine right here. This is just like a mini PCR thermocycler. And then since then, they have expanded into easy to use gel electrophoresis and other equipment. And then when I was brought on, they were looking into expanding into protein expression, which is why they brought BioBits over. And they also had the expertise for developing curriculum, which is the one piece that I was really missing. Because I learned really quickly in my prototypes is you could have the coolest hands-on experiment in the world, but if you don't have the proper curriculum guiding the teacher and the student through it, it's pretty much useless. So brought BioBits, my work on BioBits over to miniPCR, spent the first year or so there developing a company the curriculum that would accompany the labs, and then actually brought it from prototype to a real activity. So miniPCR is a relatively small company. I got to be hands-on from kind of start to finish. Not only was I involved on the science side, but also on the curriculum side, also on the product design and production side. I got to see, once we released the BioBits, we got to see it adopted in tons of different classrooms. And the pandemic actually hit a few months after we released the first BioBits kits. So we were able to really quickly pivot into creating at-home labs where teachers or schools could send students home with these little kits that everyone can do together over Zoom. Yeah, so someone's asking a little bit about the cost. So the cost right now for BioBits, obviously with like overhead and everything, a single BioBits kit that can accommodate 32 students is about $100. So that's like $3-ish per student to run a whole set of reactions. And I'll talk a little bit about the reaction that they can run. And compared to other similar labs out there, the most similar labs out there are just full-on bacterial transformation labs. That's all they have for protein expression. And those are much, much more expensive than what we can offer. So even though $100 for 32 students sounds like a lot, that is way more affordable than anything else that is on the market right now. And if you go into the papers, it breaks down the cost a little bit more if you want to see exact price per reaction. I don't remember it off the top of my head right now. So the very first BioBits lab that we released, obviously we prototyped this huge range of activities. But one thing we learned was that even though some of the labs we were developing were super, super cool, like this, the more synthetic biology, the more sensor ones, those weren't in the biology standards that most teachers teach with. And what teachers are looking for is, unfortunately, they don't have a lot of time to run labs. So the labs they run have to be concepts that they teach. So the very first lab we released was the central dogma BioBits lab. And this is one of the most important concepts in biology, but students often find it really boring because teachers just have to lecture about it. They do paper models, but there really is no hands-on lab beyond bacterial transformation, which a lot of teachers struggle to bring into their classroom. So in the BioBits Central Dogma Lab, the DNA that they add to the BioBits contains an aptamer. And when that RNA is expressed, the part of the RNA that codes for the aptamer will fold up on itself and bind a chemical, which then this binding interaction emits green fluorescence. It's how RNA aptamers work. The students, when they see green, they know it directly indicates that they have RNA. The DNA is also going to encode for a red fluorescent protein. So when it goes through translation, they get the RFP, it's going to emit red. So students are able to break down the central dogma, see green, know they have RNA and they have successful transcription, and then see red and know they have protein and have successful translation. Question about the aptamer. It's a broccoli aptamer that we are using. I think it's just the generic double quadruple loop. I forget aptamer. I think it's in our papers somewhere if you look. So that lab allows students to visualize the central dogma and really break it down. And a lot of teachers have said that, let's see if this, not sure if my video is playing. I'm in Google Slides instead of PowerPoint because my PowerPoint's not working. But that video would have it been going green to red. So sorry, the video is not working. If you are curious, if you go to miniPCR's website, we have a lot of other videos that kind of show these reactions in action. So let me actually bring up a link so I can put it in the chat and for you guys to reference. Sorry, the video is not working. Okay. And since then, we have released a lot of other labs. I'm really proud to say that the Central Dogma Lab is actually miniPCR's best-selling lab right now, which I'm very excited about. We also have a couple other BioBits labs, including one looking at protein structure and function, one looking at antibiotic resistance, and one that I just released looking at protein expression and purification, where the students get to express a mix of GFP and RFP and then separate the GFP away from the RFP to learn about protein purification for a more biotech angle. So it's been really, really rewarding to see this BioBit system start from just something I was messing around with in grad school on my lab bench to be used in schools all across the country and then all across the world. Here are pictures of students, or not, these are teachers at a development workshop learning about it in Japan. We've seen people take my slides and my videos and translate them to their native language they can teach in other schools. And then I'm going to spend the rest of the talk talking about how we actually got this up in space. So miniPCR also sponsors a science competition every year called Genes in Space. This is a science competition that is free to enter for any student in grades 7 through 12. And basically, we challenge these students to design an experiment that investigates DNA in space and applies it to some sort of space issue. And so we give them this toolkit. We tell them, okay, your experiment needs to either use PCR because our miniPCR thermocycler is on the space station. It needs to use the fluorescence viewer. One thing I didn't mention in the Central Dogma Lab is the way the students visualize the fluorescence is we have these little handheld viewers. Mine is down here. You just turn it on and then blue light emits out of these holes and then students are able to just put their tubes in and see the fluorescence out here. We have a version of this on the International Space Station as well, so they can monitor fluorescent outputs and BioBits, obviously. So when students propose their experiments, they need to include at least one of these elements. And the very first year that we added BioBits to the Genes in Space toolkit, the winning experiment actually used it. This is Celine Coquillard. She was our 2021 winner. At the time, she was a high school senior in San Jose, California. And her winning project proposed using BioBits to develop cell-free biosensors for looking at water quality on the International Space Station. So a little bit about her project. These are slides I'm borrowing from Celine from her winning proposal. Basically, the idea is what if we're able to monitor water quality of astronauts using some sort of biosensor in cell-free using BioBits, and then that means all those astronauts have to do is just rehydrate the pellet, BioBits pellet, and image it in this is the version of the fluorescence viewer we have on board, and be able to use a visual detection to conclude anything about their water quality. because right now, when scientists or when the astronauts up there need to analyze their water samples, they have to return it to Earth for microbial analysis. As you can imagine, the shipping time to and from Earth is not great. And so right now on Earth, they have to culture the bacterial strains, isolate the DNA, do gene sequencing, something that, they're kind of limited to doing on the ISS. It demands a lot of resources. And obviously, if we start doing more long duration space flights, like going to the moon or going to Mars, obviously we're not going to be able to keep sending samples back, especially if, what we're testing for is something that's going to be directly impactful to the astronauts' health, and we need to know right away. So before we can actually create a biosensor that could pick up, water quality and that kind of thing, basically we have to validate cell-free in space, because cell-free reactions have never been run in space before. And as you know, the first thing we have to do is just make sure it works in space. So we sent up a series of experiments. The first one was just looking at transcription translation, basically exactly that central dogma lab that we talked about earlier, RNA giving off the green signal, protein giving off the red signal. And we did send up some biosensors. These biosensors don't sense anything. In particular, the biosensor that senses the chemical analyte is actually just sensing the chemical analyte that goes with the aptamer. And the nucleic acid biosensor is just a random sequence. Again, to validate the system before we implement something more applicable. So for part one, looking at transcription translation, like I said, we sent up the central dogma lab. So we expect to see green after, just a few minutes. And then after overnight incubation, the red fluorescent protein. Since we got to send up experiments, we decided to also send up expression of GFP as well, just to have a second protein that we could express and visualize up there. And again, you can look up Celine's paper. I'll look at the citation here in a bit. But basically, we found that it worked in space as well. We were able to validate it. We were able to see, you can see ground versus flight. You see that the green signal gets brighter and brighter as time goes on and eventually turns to red, versus the second plasmid. We can see that the green fluorescent protein is visible the next day. We did point out there is some user training and user variability we'll have to, get across. This one, they kind of mispipetted the pipette or the pellet. As you can imagine, the pellets are going to float around in space, which is why a few slides back, I showed that there were these like little rubber stoppers. So the scientists actually have to nudge the stopper aside with the pipette tip, add the reaction, and then pull the pipette out so things don't fly around. And yes, when I say flight, I do mean the International Space Station. So these are actually these are data that was the flight data is actually carried out by astronauts. And from our quantitative analysis, because what we did is we did image saturation analysis on the pictures we got from the astronauts. And we did find that there were some differences between ground and flight. We don't think it's a biological difference. We really think it is a user-to-user, equipment-to-equipment variability. So obviously more research is needed in this. And then the second part was actually looking at some of these biosensors. This, again, is treating the aptamer like a biosensor for a chemical analyte, increasing concentration of the chemical that binds to the aptamer to see increasing fluorescent output. And we were able to show that, yes, the biosensing kinetics via the aptamer worked on flight, as same as it did on ground. Same thing with the nucleic acid biosensor. This was based on our toehold system, where if there's no target DNA, or if we give them the wrong target RNA, nothing is going to light up, versus if we give them the correct sequence, we will see red fluorescent protein light up. And we were able to show, yes, this also works in space as well. I know I'm going over this, very high level, but if you want to read more into detail, skip ahead here. We actually published in ACS in Bio, made the cover again, which is very exciting. Celine got to be first author on this paper. And again, it's open source. So it's just the validation of cell-free protein synthesis aboard the International Space Station if you wanted to read more about this paper. And these are just really fun shots from the experiment as part of the reward for winning the Genes in Space competition. We fly the winning students to Cape Canaveral to watch the launch. So that is Celine watching her experiment go up in space. I was also present. It was really exciting to see my little BioBit babies go up in the shuttle. This is astronaut Bob Hines, nicknamed Farmer. This is him actually performing the experiment aboard the ISS. And as you can see in this picture, someone in the chat is asking, do we use a plate reader? Actually, no, because again, we're very resource and equipment limited up there. So what they actually do is you can see there's this iPad that's basically attached to our fluorescent viewer. And we use the iPad to take pictures of the readouts, and then those pictures are sent back down to Earth, where we use ImageJ to analyze the saturation and use that to quantify. And the fun part is I get to wake up at 2 a.m. to talk to the astronauts and walk them through the experiment while running the ground controls at the same time. It's very, very early because that's when they're up. But it's really cool to be able to talk directly to the astronaut and coach them through running my BioBits experiments. So that was the first cell-free experiment we sent up. And like I said, in just a few weeks, fingers crossed, because launches change all the time, we're actually launching the experiment from our 2024 winners. We usually launch a year after the students win because it takes about a year to develop and implement on the ISS. This is Julia and Isabel. They were high school seniors in New York City, and they were suggesting looking at bacteriophages in space. Their idea is that bacterial infections are hard to treat in space. Scientists have noted that bacteria, for whatever reason, seem to have increased growth and virulence in space, and antibiotics also seem to be a little less effective. So what if we could synthesize bacteriophages as an alternate to antibiotics to treat bacterial infections? So they propose, and again, I'm borrowing the slides from their presentation, they proposed being able to use cell-free reactions to synthesize a bacteriophage using BioBits and then visualize it using, again, the fluorescence viewer. Very ambitious experiment, but obviously we had to simplify it. Unfortunately, we always have to simplify the experiments the students want to send up because of resource limitations and whatnot. And so to simplify their experiment, we decided to just express a single element of the bacteriophage. So instead of expressing the whole thing, let's just validate that we can express even just one part of the bacteriophage, because up to this point, the only thing we have expressed in the cell-free systems on the International Space Station is fluorescent proteins. So can we actually express like an enzyme and evaluate its function? So the experiment we're doing here is we're going to use cell-free to express an RNA polymerase found in phage and test their activity. So basically how this works out, we partnered with NEB and actually used their Pure Express system for this experiment. In this experiment, the Pure Express contains T7 polymerase, and we either added a plasmid that contained an sp6 polymerase that was wild type or an sp6 polymerase that's mutated, sp6 being a type of bacteriophage. And obviously these plasmids have a T7 promoter, so the T7 polymerase can initiate the transcription there and get you either an sp6 polymerase, either the wild type, which would be functionally active, or a mutated version that they made, which would be catalytically inactive. There's a second plasmid in this reaction as well. So after the sp6 is made, there is a plasmid in there that is has an sp6 promoter. And so hypothetically, if you have the sp6 polymerase from the expression, it should be able to act on this plasmid and express the GFP. And in this case, the wild type should be able to initiate that expression and get GFP, whereas the catalytically inactive polymerase should not. Quick question. Oh, yes. So yes, there is a plate reader on station. Unfortunately, I'm not going to get into it, but there's a lot of politics on who is allowed to use what equipment up there. So we implement and integrate our experiments through Boeing, who is our Genes in Space partner. So Boeing is only allowed to use Boeing approved equipment out there. That is true of all other equipment. So we are limited to the equipment that we have access to, which is unfortunately just our miniPCR, our fluorescence viewer, and some other equipment that does not include the plate reader. So yes, there's a plate reader up there, but we are not allowed access to it, which as a scientist makes me, upset, but that's just how it is. So getting back to this, if we are able to successfully express the polymerase, we should see the GFP light up. And so the experiment that I'll be sending up in a few weeks, we have a negative control, no plasmids added, a negative control, just the GFP plasmid added, which if there's no SP6 expressed, it shouldn't work. Then the wild type and the catalytically inactive mutant. And the only one that we should expect to see GFP expression is the wild type SP6. That's the only one that can initiate transcription and then the expression of GFP. So again, the workflow for this is we're going to heat it up in our miniPCR. We're going to use it as a heat block and then stick it into the viewer and take pictures and get that sent down for us to do quantitative analysis on the ground. So this is launching in a few weeks. Currently, it's going to be launching on August 21st on one of the resupply missions. I'm going with the students to see their experiment launch, which is very, very exciting. So stay tuned for the results. All these students publish their results, so hopefully we'll have a paper publication based on the outcome of the experiment in a year or so. So obviously, I can't do all of this alone. There are so many people at my team at miniPCR Bio, the student winners and their mentors over at Genes in Space, my original collaborators over at MIT, the VEAS, and Northwestern, and obviously Boeing for helping with the integration of the Genes in Space experiments aboard the International Space Station. And I'm happy to use the remaining time to take any questions that anyone might have. And if people have any specific questions that they want to contact me about, my email is here and I'm putting it in the chat as well. It's just ally@minipcr.com. I'm hopeful that we'll keep developing more BioBits experiments. I would love to go back to my original prototypes and see which ones are relevant to develop into an actual lab activity for teachers, and also continuing to work on Genes in Space. Hopefully there will be an International Space Station in the next decade or so that I can keep sending experiments to, but we shall see. So happy to answer any questions either in the chat or if you guys want to unmute and shout out.
Awesome. Thank you so much. I've been following those experiments really closely and I'm really excited about it. If anyone wants to chime in with questions, go ahead. But I have okay, Taesok, you go first.
So this is fantastic. I love it. Also, I'm doing some space synthetic biology. I always wanted to send my bacteria to ISF. I couldn't do so. So my first question is a more question for me. How you connect with those people that has ability to send sample to ISS? Because I just applied to NASA, but the current climate.
Yes.
So we didn't get the money, so we cannot send the sample. Yeah, that's my first question.
Yes, unfortunately, it's a lot of politics.
Yeah.
We were lucky, and this is before I joined miniPCR Bio, we were lucky in the early days of miniPCR. The founders were showing the miniPCR thermocycler at a, not a career fair, some sort of, kind of like startup mixer. And someone from Boeing happened to be there, saw it, and thought, oh, this will be perfect to send to space, because I thought PCR machines were the size of the microwave and we can't bring those up to the International Space Station, but this would be perfect. And so it unfortunately involves a lot of politics, a lot of networking. And the only way you can get experiments up there is you do have to go through an integration partner, whether that be Boeing or Northern Gunthrop, I think is the other one. Basically, one of the companies that supplies the shuttles, or SpaceX, one of the companies that supplies the shuttles and the equipment that goes up there. Unfortunately, I don't know the details of all of that. I just work on the science.
So
And
your role is nothing to do with the NASA people, it's something to do with the Boeing people.
so I have connections both at NASA and Boeing that I made through miniPCR that they already established. And basically those are the team that I work with to make sure that we pass all the safety checks and basically get manifested onto the resupply. It's a lot of red tape. It's probably the most boring and painful part of my job. And again, I don't know how those connections were made initially. There are other similar programs out there, similar to Genes in Space, that send student experiments up. And so you could always connect with one of those programs, or even connect with us. And if what you are working on overlaps with what the winning experiment was at that year, we're happy to join forces, that kind of thing.
Sure. I need to figure out that one, but I'm probably too busy to, do more. So my second question is, I really like your education effort, kind of exciting, basically, to excite young people, including high school students. That all kind of thing also requires some funding, I believe. And then I also wonder whether you have planned to extend the competition to the undergraduate student, because there are a lot of synthetic biology undergrad students, including IGEM, people, etc. Those people also could be another good people you could target.
Yeah, for sure. Unfortunately, right now, we only have the bandwidth to have the program as is with the middle and high school students. With the current political and funding climate too, we for sure wouldn't be able to expand. The competition is currently only US based, so we did discuss opening it up internationally in future years, but obviously more politics, more red tape we have to jump through. So right now we are limited to just like middle and high school, just because of resources and funding, but we would love to expand if we were given the opportunity in the future.
Yeah, at least I want to see, hopefully you secure funding. In that way, you could expand that at least at all the, US institute. In that way, that will also excite a lot of young people. And
Yeah,
that
for
would
sure.
be fantastic. Thank you so much. This is awesome. I love it.
Thank you.
Thank
Thanks
you.
for your questions.
I'm there with TASOC. This is awesome and I love it. I have a kind of a more research question. If I understand that correctly, you switched to Pure for this year's, right?
Just for this experiment, because we often, when in grad school, we would always prototype in Pure first, just because Pure Express, doesn't have all those endogenous enzymes putting around like the crude extract does. So it's easier to prototype in. And then once you, kind of get the prototype figured out, you often switch over to the crude extract if you want to make a cheaper system.
Yeah.
With Genes in Space, we have such a crunched timeline to develop the experiments that we only often have time to do it in Pure Express. And we were lucky enough to partner with NEB, who is also partial funder of Genes in Space, that they were just like, yeah, take all the Pure Express you need for this experiment. So we just ran with it. If we had more time, we would try the more crude extract, but just based on limited time, we just did Pure Express.
Yeah, that makes sense. I was mostly asking because, we work with both extract and Pure too, and I just know how terrible Pure is compared to extract yield-wise. So I was.
Exactly. Yeah, it's the trade-off, right? It's a cleaner system, but it's much less robust. But we often find sometimes that we want a less robust system sometimes because it's easier to see certain differences.
Yeah.
So it really just depends on the experiment.
Awesome. Good luck with that one.
Thank you. I just hope the launch goes off. They already delayed the Crew 11 launch, I think, by a day. So whenever one launch is delayed, it just ripples. So
Yeah.
it will go up eventually. So
Yes.
fingers crossed.
Well, fingers crossed. And thank you so much for an amazing talk. And
Yeah.
thanks for answering all the questions.
Yeah, happy to answer any more. I don't know how long you want to have me here, but I can stay as long as how many people have questions.
Yeah, I think last call for questions. If there are no more questions, I want to say thanks again. And thanks, everyone. And fingers crossed for good weather, working equipment, and healthy astronauts.
That is the big thing. Now, there won't be any astronauts on our launch, just supply. But yeah, the other day they called off Crew 11 T-1 minute when the astronauts were already all loaded inside, which is always very cool.
Sucks.
Yeah, that's always like we have a 4 a.m. launch. So I'm going to stay up all night, and it's gonna suck if they scrub it at T-5 minutes or something. So
I know, yeah. When we were doing a launch too, it was like 3:30 a.m. I think. I remember it was some ridiculous hour, and yeah, you stay up all night, and then now you're just hoping the weather's gonna hold, and it's like fingers crossed.
All right. If anyone has any other questions, you have my email. Feel free to reach out and connect. But otherwise, thank you so much for having me. These are always really fun to do.
Thank you, and bye.
All right. Take care.